Optical transducers in diagnostic biosensors. In case of optical transducers, a light source is used and that is employed for the detection of the biorecognition process. We know that when a target molecule binds with its bioreceptor element, some signals are released. And if we are using an optical transducer, it is supposed to be that we are determining the optical signals. Optical signals are signals that are produced by the change in the properties of the light wave. There are two types of light sources that are used in the diagnostic optical biosensor. One is a monochromatic light source and the other is white light. Monochromatic light sources are of two types. Number one is light emitting diodes and number two is laser. We have studied about these sources in our previous modules. We know that the white light is a combination of 7 different colors. The selection of a light source is dependent upon the type of biosensor and the transducer as well. Another important thing is the type of biorecognition signals, that what type of biorecognition signals are being generated by this process. The optical transducers are the most popular transducers among the diagnostic biosensors. Uh, Optical transducers are used in different applications of diagnostics. These transducers are employ the basic principles of optics. The first principle that is utilized by the optical transducer is refractometry. Refractometry is the method that measures the refractive index of the substance. And refractive index is defined as the ratio of speed of light in a vacuum to the speed of light in another substance. This property is used to assess the composition or the purity of the substance. When there is a biorecognition process, there is a difference in the optical properties of the wave that we are using. And this difference could be compared with the standard sample in which we have no biorecognition element, in which we have no biological target molecule. As a result, there is no biorecognition signal generated. So we can compare the two waves and can detect the presence of a target molecule by using the refractometry. The second principle that is used for optical transducer is the reflectometry. Reflectometry is the another important quality of the waves, and it is basically the reflection of the wave on the surface and interface to detect the characterize the or to detect or characterize the object. There are many types of reflectometers, and these reflectometers are used for determining this property. One type is time domain reflectometer. Time domain reflectometer is used to determine the characteristics of the electrical line by observing the reflected waveform. And this reflectometer is used to determine the uh, metallic cables if there is any defect. If there is a defect, the reflectometer will show that change. Another type of important principle is, or the technique is, ellipsometry. Ellipsometry is an optical technique and it investigates the dielectric properties of the thin film, and it can provide us the information about the dielectric function and the complex index. Ellipsometry basically measures the change of polarization upon reflection or transmission, and you can compare it with the model, and then we can detect if there is a biorecognition process. In this slide, you can see a schematic diagram of the spectroscopic ellipsometric measurement in which the linear polarized incident light that is shown in the right changes to elliptically polarized light which is shown in the left after oblique reflection of the samples. Modeling the changes in the polarization can provide us the following information about the thin film. First is the thickness and second is the optical constant. Surface plasmon resonance imaging. This is an optical technique that is heavily used in the optical transduction processes for the diagnostic biosensors. This technique is label-free technique and it provides us the chance to analyze the biomolecular recognition process in real time. The imaging is reliable and it can provide us the entire working area and the work in the multiplex format. It means at one time we can run several samples. This slide will make the concept clear. In this sensor, the gold nanoparticles that are coated with silica gold nanoparticles and silver nanoprism were used. They were immobilized on the gold surface and target DNA was detected. The gold surface was very helpful in for the determination of the target molecule. Now, the related questions: what is surface plasmon resonance imaging? And define the following terms: reflectometry, refractometry, and ellipsometry.